Hello students, welcome back to Kaplan Academy, and today we are going to discuss about cereals processing, right? So in this section, first we are going to discuss about the general feature of the cereals, about their seeds and their types, and then further we are going to discuss about the processing part, right? So let's start. So general feature if we talk about the cereals, they comes from the family called Gramineaceae, right? Which is a grass family. They produce dry seed fruits, which we actually consume as our food, right? And these fruits sometimes they are called as caryopsis, sometimes they named as kernel, and sometimes they named as grains, right? Caryopsis means that seed will be fused, not the seed, sorry, the pericarp of your seed. The pericarp of the seed will be fused with the seed coat, right? And they both will make caryopsis. This is called caryopsis, right? So if we see a normal cereals and grain structure, seed structure, then you will see there is a seed coat, pericarp, endosperm, scutellum, embryo, and aileron ray. So these parts are present in all type of the grains and they vary in their structure, right? So in this case, caryopsis means it is a fusion of seed coat and pericarp, right? And they contain degrading enzyme. The embryogenic part, it contains degrading enzymes, right? Which actually used by the seed during germination. So first we are going to talk about the wheat seed quality, right? Its processing and its characteristics, which are very important for the wheat flour processing, right? So let's first talk about the wheat, right? So as we all know, the wheat seed, it actually consists of, again, a brush which is present outside of the seed, right? Starchy endosperm, which is present in the inner core of the seed, a neuron layer, hyaline layer, seed coat, and then inner pericarp, which is called tube cells and cross cells, and outer pericarp, right? And then we have germ, which is consists of scutellum and embryonic axis. So these are the part of the wheat seeds, right? So if we talk about the wheat quality, so quality of the wheat is mainly depend on the grading level. It depends on grading level, right? Its protein quantity and the damage to the wheat, right? So the person which actually changes the seed into consumable flour is called miller, and sometimes it is called flour miller. So its function is actually to reduce the size of the seed grains into the flour particles, right? And this miller, it has only two particular goals. Number one is that it has to supply the flour of specified product to the customers according to their demands, right? And second function is that it has to efficiently separate three parts of the seed, which are your endosperm, bran, germ. Right? So Miller function is here to separate all these parts one by one so that you can get a specified product finally. Right? And the wheat is actually classified into two types. If we talk about wheat according to the pigments in the seed, it is classified into two varieties. Number one is called red wheat and second one is called white wheat. Right? So if we talk about its seed properties, so in that case, number one is your endosperm, right? And endosperm has more than 30,000 cells which vary in shape and size and also in the composition of starch and protein. So larger the seed, it will have larger the endosperm and it also have larger the starch content in it, right? So if we again look into the seed structure, then in that case, inner core of the seed, which is the endosperm part that contains high concentration of the starch, and outer core, this one core of the seed, it contains high concentration of the protein, right? So if we see in the way of nutritive way, in the way of nutrition, both of the part are very, very important, outer and inner part, right? So let's talk about now then bran. Next part is the bran. It is actually consists of pericarp, outermost tissue, and aileron layer. Aileron layer. So these are the fruit coat actually, right? And this pericarp, as we already discussed, it is fused with the seed coat, this which we call tester, which is known as tester part, right? So if we talk about the third part, which is germ, so in the germ, embryo and scutellum are present, right? And they are separated from the endosperm. They both are separated from endosperm. How? By a layer of epithelial cells. By epithelial cell layer. Right? So embryo actually takes its nutrients from the endosperm by epithelial cell layer. So let's again see the image of the seed. So we see that here is the layer of the epithelial cells present in between the endosperm and in the embryo, and the nutrients comes to the embryo through this epithelial cell layers, right? And during the germination, it also activates the enzyme, right? And those enzymes are degrading enzymes. During germination, certain degrading enzymes are actually activated, and these enzymes include proteolytic enzymes, they will lyse the proteins, and also amylolytic enzyme. So they both will actually break the complex structures into the normal small structures, right? And they help in the production of sugar residues. Next, if we talk about the quality parameters of the wheat, so number one parameter comes is the moisture. Wheat quality parameters, right? And in this case, number one parameter is the moisture. And these parameters are taken care during the every type of flour processing. So first parameter is the moisture, right? It is not a factor, it is not taken as important factor by all the countries, but some countries actually involve this factor also. And according to them, the moisture of the seeds and flour should be optimal. So if we talk about the seed moisture content, it should be within 14 to 17%. And if we talk about the flour, it should be 14% at 25 degrees centigrade, and it should have 60% humidity, under 60% humidity, right? So by this, moisture is a very important one, and this content varies from countries to countries and region to region. Next important parameter of the wheat is the wheat ash content. Wheat ash content, right? And this one is expressed as percentage of original sample. Original sample, right? And if we try to define this, then it is a residual inorganic material left after the incineration of the flour. So rest of the content is called ash after burning of the wheat flour. So it is used to evaluate wheat flour qualities. Wheat flour qualities, right? And it should be under 1.5 to 2.00%. So this is the range of the wheat ash which should be present in all type of wheat flours, right? Another parameter of the wheat seed is that kernel size, right? Change in size, seed apertures, fixed configurations can affect the grinding performance, right? So the size of the kernel, it should be uniform. If it won't be uniform, then it can affect, as I already mentioned, it can affect the processing of the wheat during milling, right? So the behavior of the different sizes of the wheat kernel in the milling, it was studied to influence the size on water absorption. Water absorption. And it is done during the tempering and break release. During the tempering and break release of the seed. Right? Next parameter comes, that is kernel shape. This was the size, and next parameter comes, that is kernel shape. So this one is actually used in the classification of hard or soft wheat. So this one is also very important because this will also can affect the grinding processing during the milling, right? Next thing come, that is kernel hardness. And this one is used for the quality and suitability of grinding the seeds during milling. all these parameters need to be taken care during the processing of the seeds, right? Another parameter is the protein content. Protein content, which is very important because without the nutrients, your flour would not work for anything, right? So here the quality and quantity of the protein in wheat, it is adjusted by wheat mixing. It varies from one particular type of the protein variety to the another type of protein. Sorry, and one particular of this wheat variety to another variety of the seeds, right? Another thing which is used to actually, another important parameter which is also used to check the quality of the wheat flour is the falling number value. Falling number value. And in this case, what happens that seconds, time in seconds required to stir a hot aqueous flour, which is undergoing liquefaction. Liquefaction, right? And it is going on in a viscometer. To this process, and then the viscometer stirrer is allowed to fall in the test tube in certain distance. So if we see in an image which I have for you here, that in this case, that first you actually stir your flour at high temperature with the water, and during this process you stir it with a viscometer, right? And at certain time period, you actually allow the viscometer to fall in the test tube, and that distance is measured through the gel, and it tells us about the quality of your wheat flour, right? It will tell flour water absorption quality, water absorption quality, and it also tells about the duff handling problems, right? Other than this, the absolute minimum falling number, absolute falling number for the floor is actually calculated within seconds. So for 60 seconds, you actually measure the fall of the viscometer, right? Another parameter is the floor yield. Floor yield, right? And this one is very, very important because it actually tells you about the buying decisions of the customers. Buying decision of customers. So if the customers, they actually not satisfied with the yield of your crop, then they can reject the produce regardless of their other characteristics. So this is also a very important factor during the wheat processing, right? Another variety of the, so these are the parameters of the wheat which actually need to be taken care during the food processing, right? Another variety of the wheat which is very popular and used for the production of various type of food material, that one is called durum wheat, right? It is red in color because of the seed coat pigment, right? And it produces granular endosperm product. It produces granular endosperm product. and that one is called semolina. It is also used in the making of pasta products, right? Why? Because the durum wheat seeds are actually very hard and they are very hard to process into flours. So because of that, they are used for the production of other type of food material like semolina and pasta, right? Like you can see in this one picture, right? So now this was all about the wheat seed. So the same properties and parameters are used for other type of cereals too during the cereal processing, right? So now let's talk about another type of seed or grain, which is rye. So in the rye, it does not have any hull part, no hull part, right? It does not have any glumes, hairy structures on the one tip of the seed, right? And it also has caryopsis. It has caryopsis, which means seed coat will be fused with the pericarp in it, right? And it contains then pericarp, seed coat, nuclear, nuclear, nucellar, epidermis, germ, and endosperm. As you can see, aileron layer, sub-aileron layer, endosperm, right? And it will have its content, protein, starch, and cell wall. So this is about the structure of the rye. So in this case, starch is actually embedded in the protein matrix. So starch and protein, they will be very close to each other, right? Next seed structure is the rice, which is also very important. So rice is also basically same as other cereals, but it has some difference from the others, right? So first of all, it is harvested with hull or husk, which is attached to it. As you can see, the outer layer of the seed coat, right? And when there is a hull or husk attached, those are called paddy or rough rice. Rough rice, right? And in this case, hull is actually 20% in the seed, right? Other than this, it contains lemma, palea, right? And the composition of the hull is that it contains lignin, cellulose, and hemicellulose structures. Hemicellulose components, right? So brown rice, if we talk about the brown rice, so it is the same structure as other, but it does not have crease, right? So your brown rice, it actually does not have crease structure in it. So as you can see in this one, it has a different apex, which is called on structure. It has lemma in the hull, right? And then it has a bran, germ, which contains the embryo, and then the endosperm consists of endosperm cells containing the starch granules and aileron cell layer, right? So now let's talk about another seed variety, that is the oats. So oats also, they have same caryopsis in their structure like others, and they are called groat. They are called groat and covered with trichomes. As you can see in this image, these are hairy-like structures. So they are covered with trichomes. And these trichomes are not present in other type of cereals which we discussed. Right? Then let's talk about the corn seed. So corn seed actually, it is also have caryopsis, but it is dented seed. If you see its structure, it is dented one. and it also contains the same pericarp, right, which is tube cells, cross cells, mesocarp, epidermis cell there. Then it has seed coat, which is known as testa. Then it has the germ embryo, tip cap, and endosperm part, which contains the starch content, right? So if we recall all type of the seeds, they have the common structures in their seeds, and those are embryo, endosperm, and pericarp, right? They are different in certain types of the seed structures, but most of them they have these common features in them, right? So this is all about the seed structures and the general properties of the greens, right? So in this section you need to remember about their seed structure because it is very important. So sometime question can be asked from it, right? So in the next class we will discuss about the processing part of the cereals. So that's all for the today. Thank you.